Hello, welcome. It is about 11 o'clock in Madison, so we're going to get started. Welcome to everyone. Thank you all for joining us today. Good morning, good evening, whatever time of day it is where you are. We're so glad you could join us. I'm here today with my colleague, Matt Kaiser. Matt is our Senior Product Manager, and we are going to show you the new features in Lasergene 17.1. We released 17.1 about 2 weeks ago, and it's got a lot of new features, a lot of improvements to things that were released in Lasergene 17. So I think you're really going to like what we have to show you today. Just a few housekeeping items before we get started. We want this to be an interactive session. We want to answer questions that you have, address any comments or feedback. So if you do have a question, use the questions tab on the right. You should see a little go to webinar toolbar, so you might have to expand that in order to see the questions panel. But if you type in those questions, those come to me, and then I'll ask Matt to answer those during the webinar. And a question we often get is whether or not the webinar will be recorded. Yes, we do record the webinar, and we will send you a link to that recording in the next several days. So just a quick overview of DNASTAR Lasergene in case anyone here is not familiar. I know we have some new users with us today. DNASTAR Lasergene is a comprehensive software suite, and so it consists of applications for molecular biology and sequence analysis, next generation and now long read sequencing, assembly and analysis, as well as structural biology and protein analysis. And all of these areas had updates in Lasergene 17.1. Today, we're going to really focus on the molecular biology features. And then we have another webinar scheduled for August 5th, where we'll cover some of the new genomics features. And I know Matt's going to show some of those today, but just a heads up that today's going to be a pretty deep dive on the molecular biology and just a higher level overview on the genomics features. So as you'll see, as Matt's showing the software, all of the applications have a really nice interface. We try to make the software easy to use. That's part of why we have these webinars. We want to make sure people see the new features and have a chance to ask us questions firsthand. And also we provide highly accurate results, and that's really seen in just the widespread use of our software. Lasergene's been cited in over 80,000 publications. It's used in most academic institutions, so it's really well-known software. If you haven't used Lasergene yet, we do offer a free trial, so you can also get a fully functional trial and see this for yourself and walk through some of the things Matt's going to show you today. So what's new in 17.1? A lot of new molecular biology and sequence analysis features. So we've really beefed up our SeekMan Ultra application. SeekMan Ultra was introduced with Lasergene 17 in February of this year. And we've added some really nice enhancements to the program to allow for easier assembly of your data, multi-sample variant analysis, as well as some other visualizations and enhancements. Another big area of improvements in Lasergene 17.1 is multiple sequence alignments. We've made improvements to being able to edit your alignment, being able to trim sequences within an alignment, and also some improvements for phylogenetic trees. So we've added RAxML for maximum likelihood trees. We've added bootstrapping. Matt will show you those features as well. And then we also added a batch cloning workflow in SeqBuilder Pro, so you can clone multiple inserts or a single insert into multiple vectors and really streamline your cloning process. We've also got some improved exports of analysis tracks in Protean 3D. But really on the whole, some good features for automating and better visualizations of your data. as I mentioned, SeekMan Ultra was new in February of this year. I know many people haven't updated yet, so this might be your first introduction to SeekMan Ultra. This application replaces SeekMan Pro for assembly and visualization. So we will be retiring SeekMan Pro in the future. So if you haven't started using SeekMan Ultra yet, I definitely recommend you start using that application now. All of the new development for assemblies and visualization is going into SeekMan Ultra. SeekMan Pro is not being actively developed. So just something to keep in mind if you're still using SeekMan Pro. And SeekMan Ultra uses SeekMan Engine for assembly. So if you've used DNASTAR software for next generation sequencing, you might be familiar with SeekMan Engine. That's being used now to assemble all data. So whether you have Sanger data, next gen sequencing data, or long read data, all the assembly happens in SeekMan Engine, and then the visualization happens in SeekMan Ultra. So also new in Lasergene 17.1 are genomics features. So we have a new retrovirus integration workflow. I think Matt's going to show you how to access that today. We won't go terribly in depth with that. There's new options for combining multiple projects into a single project. And then also de novo assembly of long read sequencing data from Oxford Nanopore and PacBio. And we've got some really nice workflows for doing genome finishing, different kinds of de novo genome assembly strategies. So whether you're doing purely de novo or using a reference to guide the assembly, or using short reads to clean up a long read assembly, we have a lot of options that can help you there. And so that is going to be the focus of our next webinar on August 5th. So if you have long read sequencing data, or if you're doing any kind of genome assembly, I highly recommend you join us for our next webinar as well. So with that, I am going to hand it over to Matt, and he is going to do a software demonstration.
Thank you, Katie. And just a moment here and I'll show my screen. And I think we're showing here.
Yep, it looks good.
Great, great. Well, thank you again, everybody, for joining us today. I know that we're covering a lot of different topics, and I'd be happy to take questions, as we kind of move from topic to topic. Katie will let me know if there is a question, and so I encourage you to ask questions. And if we don't get to your question during the webinar, I'll do my best to get back to you later today or tomorrow with any questions that you have. And so, as Katie mentioned, we've made a lot of improvements to our molecular biology tools and our genomics tools, and we'll go into some detail, and I'll show you the new interfaces in the mobile tools. And then in August, I'll have another webinar where we'll really dive into some of the genomics tools. But I will show those to you today so you can see where they are in the software. So I've got the DNASTAR Navigator open, and we've made some slight changes here to try to streamline it a bit, but we're going to start with some Sanger ABI assembly and analysis. So by clicking on the Navigator, I can launch specific workflows. There's also a little organized by application link that would allow me to show the application list. So if you're familiar with the different applications, of course, you can just select it from that list. Let's make this go away here. There we go. So SeekMan Ultra, as Katie mentioned, is the replacement for SeekMan Pro. And we've been working on it for, a year and a half now. We had a release earlier this year. That was the initial release, and we continue to develop and improve SeekMan Ultra. And it's going to have a similar look and feel then to some of our other applications like Protein 3D, MegAlign Pro, GenVision Pro. Will have a very similar look and feel, and to some extent SeqBuilder Pro as well will have this kind of similar layout. And so you can see that with Ultra, I can start a new assembly. I can also open existing assemblies. And so this is very handy, for navigating from project to project and for doing different things. And you can see that under new assembly, there's different categories: molecular biology, genomics, transcriptomics. And for this demo, we're going to focus on some of the Sanger and molecular biology. And what this is going to do, so I'll show you some new functionality we have with Sanger ABI reference guided. When I make this selection, it's actually going to open up the SeekMan engine wizard, which will guide me through an assembly. And so earlier on, if you're already familiar with our software, this was primarily a NGS assembler. Well, as we've had some focus on long reads and developed some new algorithms there, it really became apparent that we really want all the technologies to have the same benefits of going through the wizard. And that means, you know, some of the really new things that we developed for genomics are now available for Sanger users. You can align to a genome template package. You can have a probabilistic SNP caller and get the best possible SNP calling done. So there's lots of extra features now that our Sanger users have access to. But I will show you two different ways that you can use this wizard. So I've launched a reference guided Sanger assembly, and it's going to ask me for a reference sequence. In this case, I'm using some data. It's some plant genome data from a chenopod. One of my colleagues, one of his PhD work, donated some sequences for us. So we have a reference sequence here, and you can see there's this button down in the bottom right corner called Run. It's grayed out yet. I need one more piece of information before I could actually assemble this. I need to load in some data. So I'm going to treat this initially as a single sample. And even though it's not a single sample, this is these files here are you can see the 2, 6, and 8. They're forward and reverse files from really 3 different amplicons. So you might choose, you can still analyze it this way. I can align these 6 reads to the reference, and for some users, that's all you need to do, right? And you can do whatever analysis you need just by aligning them together to a reference. And you can see I have this Run Now option. So I can, in 2 wizard screens, if I want, start assembly. And because I chose this shorter version of the wizard, this kind of Run Now, I'll be able to open this. This will open immediately in SeekMan Ultra. And so when it's done, it loads the assembly in, and we get a little summary then of the assembly, the coverage and number of sequences and so forth. And I can navigate this now. So this is the summary page. What's important to know, like SeekMan Pro, SeekMan Ultra has a list of it might be contigs if it's de novo, or the reference sequences. And that was the project window in SeekMan Pro. The equivalent to that is this Explorer panel in SeekMan Ultra. And what I have selected in this Explorer panel is really a key to focus the different menu items. So you'll notice that it comes into view, and as a new user, you'll want to always remember, if I want to direct my analysis to a particular sequence or contig in my project, to have the Explorer panel in view. So this is just a small project, a single reference sequence. There are some icons here to the right that allow me to open different views. So this top view is the alignment view. Then we have a strategy view, and then there's a feature table, a variant table, and reports. So these are quick icons, then I can click on them to get my view. And I can go to, so if I want to do variant analysis and look at the alignment view, I can click those quick icons. It splits my view now into 2 windows. Again, depending on how you like to do your analysis, I actually prefer my SNPs to be in a table. I'm just used to it being below and my alignment above. And so you can click on the tabs and move the views to customize it. There is, I'll point out, as long as I'm showing that, we're going to have in the next version some more advanced tools for setting default user styles. So as we continue to work in SeekMan and SeekMan Ultra and some of the other applications that are similar, we're going to have a little bit more sophisticated ability to save default type styles. So that's something that will be coming soon. So now I have a SNP table. You'll notice that we don't see sequences up here. We have, and the reason for that is the reference in this case is quite a bit longer than the individual. It's 132,000 bases. We have a length in the middle of the window. So I could use the SNP table to find the variants of interest, but I can also use this search alignment icon. So there's this icon that then allows me to move through the assembly to find something of interest. So if I go to the first variant, it will advance then to that first variant. And now I can move through the assembly from variant to variant, and I can see some are filtered out. So we have these red icons with a diagonal that goes through them. That means they got filtered by one of the filters that are applied. And then I have the blue variants that are called variants that met my filtering criteria. These expanding icons allow me to look at features, for example, or I can look at the trace data. So if I want to see the trace, I can click on it, or I can, right-click, expand all, and see all the traces. Right. So for a small project like this, it might be okay just to have everything aligned together in one project, even though it's from different amplicons, and I can do my analysis. There's also filtering, and I won't go into great detail here, but you can see a whole set of filters. Again, because we've gone through and used the engine assembler, and it uses the updated type SNP caller. So that means that we have access to this much more sophisticated set of SNP filters, right? So depending on how well annotated your reference is, you may have quite a few extra filters now that you can use as criteria to apply filters. Okay, so that's just a project where we have all the data aligned together. So I'm just going to close this, and let's try that again. And this time we'll treat it as multi-sample. Okay, so same workflow, same reference. And I'm going to choose a multi-sample project. And this is a small project, so it's good for a demo. But you could imagine this could be, it could be a couple hundred files. There's no real limit. It can be quite large in the number of files that you have. So now we get some added text, and it's trying to guide me. So what's happening now is we've told the wizard that we've got multiple samples here. So it tries to read the name first and assign an experiment name to it. And depending on how the files are named, it may be successful or it may not be successful. Because Sanger files are so variable, the names are arbitrary, it usually can't get it right with Sanger data. With things like Illumina sequence files, they have a consistent pattern, and those are fairly easily parsed automatically. So in this case, it says, use the group selection tool to automatically group the reads. And so down at the bottom is a group selection tool. So I can look in the name here and find the unique part of the read and just select it. And now it can name my experiment. And you can see that the top two now go together and the middle two go together. Or I could do it manually. If there are some cases where it's just it can't work for all the files, and you can always select, and then group them and name them manually. So this is just a nice way to do it automated, especially if you have a large number of samples. So now, because it's a multi-sample experiment, I'm going to go through more pages in the wizard. So I have to give the assembler a little bit more information now because it's a little bit more sophisticated type project. And you can see that as I do that, there's assembly options. I can change the minimum match percentage, right? The default is at 90%. I may, I can change this, raise or lower this, right? And that's probably the single most impactful parameter. Just keep going here, and you can see it tells us what type of analysis options are happening. So we're gonna, by default, run a SNP detector, a diploid SNP detector. There are little pop-ups, tooltips that will explain some of these in more details, and of course, a help menu. And now I'm just gonna call this and pick an output folder. And so it's a little bit, a couple more samples. It'll take just a little bit more to align it. And now it doesn't auto open. I didn't use the shortcut version. So now I go to the next page and it tells me, gives me a couple different options. And so I can open the assemblies. I can compare a variant. So if I have a genomics license, I can go to RASTAR and have a pretty elaborate comparison between samples. In this case, I just want to go and look at my assemblies, right? So here it's multi-sample, and you'll see when we go to the alignment view, and I'll navigate here to the variant. Now we have this what we call a pseudo consensus, and the pseudo consensus is for each sample, and I can collapse it all down. So now I can see, you can imagine if you had a large stack of samples here, I could compare and see, how similar or different are these samples from each other. This particular data set, there's a lot of variability between the samples. Some other data sets, there might only be, you might look at a large number of samples and only one or two have a variant of interest in them. So really depending on your workflow, this might be a better approach, or assembling them separately and analyzing, or just lumping them together into a single project. So I get this pseudo consensus, and I get a good feel for, it looks like these three are quite different from each other. Doesn't look like there's anything that they share between each other too much. But then I can drill down as well. So if I want to look at a particular sample, this twisty will now allow me to look at the two traces. So for sample 8, I can now expand the traces and look at the traces for that sample. The variant table also will have a column that gets us a particular sample as well. Oh, I think I double clicked on something there too fast. So you can see, you can see over here in a sample column, I can group by samples as well. And okay. So I'm going to go back here to this assembly log. Oh, excuse me, run assembly project. So I kind of skipped over this page a little bit, but I did want to point out that if you're doing reference guided assemblies, if you're aligning to a large reference, if it's something like a human chromosome or something large in size, the wizard will assess your hardware and make recommendations. And so, for example, if I'm on my laptop and I want to align some, it might even be just Sanger data to several human chromosomes, there's a certain amount of disk space that's needed. And so the wizard will look at your data that you're adding, and it'll look at your hardware and make a recommendation. So either run this on the local computer or to run this up on the cloud. And so when you run on the cloud, so most Sanger assemblies can be run locally, right? If it's de novo, you can only run it local. They're all small enough. Most reference guided, but there are some, and depending on your computer, where you'll have the option to run that up on the cloud, and that's seamless. It will zip the data, upload it, assemble it, and download it back to your local computer in an automated fashion. So it's a nice piece of functionality there. So that's the new functionality that we have in SeekMan. If there's any questions, I'd be happy to take them on SeekMan before we move on to MegAlign.
Thanks, Matt. We do have a few questions. The first comes from Elizabeth, and she asks, if I began a reference-guided Sanger analysis from Engine Wizard, would I choose variant analysis whole genome? And would the output be the same if I had started in SeekMan Ultra with a reference-guided workflow?
Yeah, good question. So if you choose a variant analysis, it uses the same from either location. It uses the exact same assembler. So one is just shortened to make it more streamlined for Sanger users to cut it down to a couple wizard pages, whereas the other is guides you through the full wizard, but the results should be identical.
Great. And then another kind of workflow question, how does choosing multiple samples or multi-sample in a single assembly differ from choosing single sample? I think on the engine input sequences screen or in the assembly.
Right. So when it's a single sample, so the assembly is the same. So it takes the data and aligns it to the reference. What's different is that if you select multi-sample, it will create those pseudo consensus and allow you to group the different samples group the reads to their respective samples. But the actual assembly of the data is the same.
Okay, gotcha. And then this last question, I'm not entirely sure I understand. This comes from Sarah. She would like to import a bunch of viral FASTA sequences to analyze SNPs across the genome relative to a reference sequence. And she says she could import them aligned or unaligned. Is it possible to do this in SeekMan Ultra or another package, i.e. without starting from the raw sequence data?
Yeah, if you have viral genomes and they're fully intact, full length, and aligning them, they aren't really sequence reads, right? They aren't. It's not raw sequence data getting aligned to a template. So that really algorithmically is a multi-sequence alignment, and it's probably better suited for MegAlign Pro, which is what we're going to look at right now. Now, if it's a virus, you're probably not looking for big rearrangements. If there are rearrangements, then you'd want to use something like the MAUV aligner in MegAlign Pro, so it can account for those rearrangements. If it's just SNP differences, really the best option is to align in MegAlign Pro, which will take the entire viral genome and compare them to each other and make that alignment. So that would be my best recommendation versus SeekMan.
Okay, great. And that's actually the last question. So I guess that's a perfect segue to the next part of your demo.
Yeah. So we'll go into MegAlign Pro. And again, I'll just start from the navigator here. I'll switch over to applications so you can see, again, by workflow or by application. Demoes, people are new to the software, typically start by workflow. Once you're more familiar with the different programs, and you can start here by the applications. Okay, so MegAlign Pro, again, like SeekMan Ultra, has the options for new project, open a project, and search and training. So again, there's more consistency between the applications, much like the original classic DNASTAR applications had that consistency. And this is kind of the new UI. So you'll see once you learn one, it'll be similar when you move to another. So we're going to start a new project here, and I'm going to add some sequences. And these aren't super interesting sequences. They're just some exons from that are fairly well conserved. We will have, I should also point out, we will have later this year, I think in August or early September, a focused webinar as well on MegAlign Pro. So if I just lightly touch on some of the things, we'll be hitting this again in quite a bit more detail. So MegAlign Pro, I can align DNA or protein sequences. And some of the new things that are added here allow us to make some edits to the sequences or edits to the actual alignment. One common thing to do is, things like changing the name. So we'll look at these names as they get parsed out of the GenBank file aren't great. So we can do a rename. And so I can manually rename them, which isn't bad for this for few sequences, but I can also go in and see what fields are available. It varies depending on the source of the data. In this case, the file name has a pretty good descriptor. So it's a way to make the names consistent so we can recognize them more easily. So we can rename, that's been in there for a while. What I want to show you here though is that one common way to edit sequences is to say trim them back to a common starting point. And so for these sequences, an ATG, when it's an exon, an ATG can often be a good starting point. So I can select a sequence, and there are toolbar icons to trim sequences. And it brings up a dialog that is what you'll also see this in Seek Ninja. And I can trim to the ATG. And I can do that also through a context menu. So again, a common approach is to just kind of manually trim. And for some algorithms, having the common start point makes quite a bit of a difference for how the sequences align. Not all algorithms, but there are some where they're a little fussier in terms of the very start of the sequences. I know one of like the ClusTLV from years ago, that one was very fussy with the start. I think ClusTLW is still fairly particular as well. So I'm going to trim another just to show you that. And I think this ATG is in a little deeper. Oh, missed it. There we go. So a little end trimming, I could trim the other side of the sequence. I could also align the sequences and then decide to trim. Sometimes one sequence is a bit of an outlier and it needs a little bit of cleanup. But this is a way just to do a little trimming prior to alignment. So I'll just align now using ClusTL-Omega. And so we've got, many different alignment algorithms, ClusTL-Omega, Muscle, ClusTL-W, MAFT. So, you can choose the algorithm that suits your needs. There's also, and again, I won't go into the parameters here, but there's different parameters that you can adjust for these different algorithms as well. So we'll let this align. And a difference now that you're going to see with MegAlign Pro version 17.1 is that the tree doesn't automatically get generated. In earlier versions, there was only one tree option. It used the nearest neighbor joining method. And that tree was done automatically. So you would run the alignment, and then that phylogenetic calculation would occur right after the alignment finished. And so you'd have a tree ready for you. So now there is, when you click on tree, it's going to tell you which type of a tree do you want, a nearest neighbor or a maximum likelihood. And so when I click on nearest neighbor, this is a very fast tree for just a couple of sequences like this. It's probably it might be accurate enough. As you start to have larger projects with more sequences in them, proteins in particular, these trees generally aren't accurate enough for publication. They're just kind of a quick and dirty tree. So you can see now that when I've made this tree, there is an Explorer panel. I'm going to expand this just a bit. In MegAlign Pro now. So this is new for MegAlign Pro. And that is, I have the ability now to calculate different trees, to make changes to my alignment, run different aligners even, and then build up a list of trees in this Explorer. And so I could go back, for example, I could go back to my sequences, and what's kind of popping out at me is I see another ATG a little bit interior here as well. I might decide, well, you know what? I picked the wrong ATG for those first sequences. I want to trim back to this common ATG. There are a couple ways to do it. There is, I could select that region and trim it, or I can grab these. They're pretty subtle, but if I hover over them, I can trim the alignment now just by dragging this. And now I've trimmed all the sequences in the alignment to that common spot. Likewise, on the other end of these sequences, there's also the ability to trim over here as well. But I won't trim all that right. Let's expand this a little bit. so I could make a trim on both ends if I decided to. So I can make edits to my sequences and also edits to the alignment. And now I can recompute. I could say, well, I'm going to make a new tree now. So I can go back to my tree view, and there are now different places I can initiate this. Let's have to move my webinar here a little bit. So there's this little compute phylogeny button. When I click on that, it brings up an interface, and I can say I want to change this and do a maximum likelihood tree. And I can rename it too. I could call this a RAxML tree to try to keep this straight. Let's see, it was Clustel Omega. I have the option to bootstrap or not. And so for this first one, I won't bootstrap. I can pick the number of seeds, and I can also choose the number of threads. I'm on my laptop right now, and this is a tiny project, so 2 threads is more than enough. And I can click OK and get another tree. So now I have 2 trees. I have the BioNJ nearest neighbor tree, and I have a RAxML tree that's visible. So I can start to build up trees, and you'll see also there's a jobs list down below. RAxML may take, if I have a large project, and the largest that I've run thus far is about 2,000 sequences, and that took a couple of hours, to do both the alignment and the RAxML analysis. If you add bootstrapping, that will add some more time. So it's good to have a job, then I can watch for something that's long running or potentially long running. We can keep track of that in this jobs panel so that jobs are completed or still running. So again, I can go back to the compute another, turn on bootstrapping, name it. And you kind of get the idea now. And change the seeds, the iterations of bootstraps. Again, we'll have a detailed webinar that my colleague Brian Walsh will do, where he'll explain this in more detail, the phylogenetics of it and the implications. So I can build up different trees now. The trees, if I go to a details panel, as I select them, it updates the information. There's also this toolbar that's been greatly enhanced with MegAlign Pro version 17.1. And for example, I can go and rename the trees. There's a style panel, so I can change, the styles. If I pick the bootstrapping tree, I can show labels. This is distance labels. If I go to the bootstrapping tree, I can show the bootstrap value on the tree or the distance values. So I can really start to customize how this tree looks. I'm just kind of clicking on things here, so you can see I can change fonts. Ooh, that's not very good. I can change the fonts on it. I can add a box. Let's do this fast. Oops, no, I don't want to do that. I want to do there. So you get the idea. So we can make the tree look, change the appearance of the tree to make it look nicer. We have ongoing work with trees, and so later this summer you'll see ability to do things like apply styles to specific branches, select branches, reorder things in branches, that sort of thing. So again, ongoing work right in this area as well. There's also a style panel, and the style panel then is divided by the different types of views that we have. So I can change the layout of the tree, if I want over here. So there's a little bit more you can do here, but some of it's duplicated from what I have from the toolbar as well. There's also, so that's a style toolbar. I can hide that again. There's export data options here. So you can see I can export as a NEWIC tree. I can also file, add trees. And so I can add trees in NEWIC format. And NEWIC format is a pretty standard format, but the extension is not standard. It can be .tree, .text, .NEWIC, some other things. So you can bring trees from an outside source into Megalign as well, compare them. There's different views. You know, this little icon in the bottom right corner is very handy once you discover it. And you know, I can do things like this is getting a little bit crowded on my little laptop screen, but you can split the screen. I can put two trees, you know, next to each other, so you can compare trees. So the way that the views are handled here really makes it conducive then to, you know, making kind of comparisons like that. So it's a lot of flexibility with how this is viewed. Now, there's also the ability to export the image. So once you get an image that you're happy with, this is with these newer applications, the ability to export to PDF or directly to a PowerPoint or bitmap images is very handy. PowerPoint is nice because the different lines will be identified as objects, so you can make edits to specific lines pretty easily. So it's a nice ability to go right into PowerPoint with that. And then lastly, there's also these quick details, which gives us some information exactly about, you know, the likelihood score and the different options that were used in RAxML. So a lot of work here, still ongoing work with trees. And I guess there's one other thing I did want to show is that the sequences menu also has toolbars. This toolbar has tools for exporting data. So this is an alignment now, so I can export the sequences, the aligned with or without gaps, export all the data, you know, and then I have a Nexus file where I can export the entire alignment. So some quick options and to export data out. I think I don't think I'm forgetting. I think that's all I wanted to cover with Megalign. If there's any questions, I'd be more than happy to take them regarding Megalign.
Thanks, Matt. We do have a couple. The first one comes from Khadija. I hope I'm saying that right. Asking if this approach excludes mobile genetic elements. I assume they're referring to the alignment approach.
Yeah, I'm not certain. I don't think that it does, but I'm not 100% certain. That's something that we can talk with developers and see, and some of the scientists to see what that would entail.
Sure. Khadija, maybe we'll follow up with you after the webinar and get some more information on what you're looking for there.
Right.
Another question comes from Ramesh. Can Megalign Pro support alignment of whole genome sequences from bacteria?
It can, yeah. So that would be done under Align, and there's a MAUV algorithm here. And so I don't have whole genomes loaded here, but you know, you can add whole genome sequences. I don't think I have any ready to go on this computer, but yeah, it is just add your genome sequences and choose the MAUV aligner, and it can align those.
Great. And yeah, as Matt mentioned, we'll have some other webinars on Megalign Pro. We did one a couple months ago. I don't know that we got into MAUV, but we can certainly show that in our next Megalign Pro webinar, which will probably be in September.
Yeah, that might be nice to show that.
Yeah.
And one thing about MAUV is that it is up here as a workflow. So under Genomics, and this is new for 17.1 as well, we move that workflow right to the surface. So I would anticipate, you know, before it was a little bit hidden. You had to go to Megalign Pro and, you know, find it under the Align menu, but now it's right on the top. So I imagine we'll get quite a bit more feedback.
Mm-hmm. And then this last question comes from Assam. And I'm not sure if this applies to Megalign Pro or SeekMan Ultra, but you probably will know. Can I determine locations of mutations compared to a reference?
Yes, yes. So that is in SeekMan Ultra, and so the variant report so when you do a reference guided alignment, it's aligned to that reference, and it should all be in reference coordinates. So that variant report then has the reference position of where that variant is.
Right. Okay. Thank you. I think that's all the questions for now.
Okay, great, great. One more molecular biology tool. This one's a little bit shorter to show off here. It's in SeqBuilder, and it is batch cloning. And so this is just the ability to, good. The ability to clone multiple inserts into a vector, or it could be multiple inserts into multiple vectors. It is accessible through the navigator. Also under the tools menu, there is a batch clone with some of the other batch kind of operations. These are some newer functionalities that we've been adding to SeqBuilder the last year or two. There's also under the cloning menu, a batch clone option. And when you launch batch clone, it opens this cloning project window, which is really a wizard, not a multi-page wizard like the SeekMan engine assembler, but just kind of a single page wizard that gives you some guiding text here in red to try to guide you a bit. But I'll show you how I typically use this. I'll usually open an empty project and then import the sequences that I want to clone. So in this case, I'm just going to pick these 3 genes in GenBank format. And so now I have inserts that I'm interested in. And now when we go back over to the wizard under batch cloning, you can see that a prefix is going to be added to each one of the clones. By default, we have this ligation independent method selected. We also support gateway and PCR directed restriction inserts. So 3 different methods for batch clone. I need to add a vector. I could import a vector into my folder, right? And then you drag and drop them from the project, or I can choose a vector from the list that's built into SeqBuilder. So I picked just a pgem vector, and I need to linearize it. So I can use these pull downs, and what these pull downs are is it looks at the vector and the inserts and tries to pick a set that is compatible, a set of enzymes that are compatible. So I could pick Echo R1 and BAM, right? Or if I click on options, it's going to open the vector, and I could go in and manually select the enzyme. And it's kind of hard to see at the bottom of the page here, my screen, but make this bigger for you. So, or I could manually go in and pick the enzymes that I want to use. So I guess I could just do like, oops. So there's 2 enzymes. You can see if I select them, it will pick the ones that are, in this case, I just got the Hindi. So doesn't like the other one. So it's telling me that it doesn't like the BSA X1. So it'll give you some feedback to not use particular enzymes that are not compatible. I typically just use what's in this sites area. So now I can pick the inserts, drag them in, and it says it's ready to clone. It's also going to export a file of primers. So because this is ligation independent, there are going to be primers generated in order to make these clones. So it's going to generate those for me. And now it's going to make 3 clones. Again, save that file out. And now it's doing all the in silico cloning. So the end result then is a project here that's got, I'll just expand everything, my 3 inserts, the vector that I used, the ligation independent cloning inserts, the clones that were actually made, and then it also makes a file of just the primers that are kind of concatenated together. Some users might use this file. I can open it. because you can make a primer catalog out of it right from this file. So I can get that file, and it's just a file of primers. More users, I think, will probably actually use the file that was saved out. So here's my 3 clone sequences and the file that got saved out. If I go back to my desktop here, I picked my folder, I missed my folder. So it's this file. So we get this FASTA file of those primers to make those clones. So pretty easy to use. You can load in a large number of insert sequences. I don't know the exact upper limit, but it's it's going to handle I think most people's needs in terms of certainly several dozen sequences, maybe even a few hundred sequences at a time. Now I can also just do one more kind of variation on this. So again, batch clone, bring in my inserts. And now I can also pick more than one vector. So I'll just pick a pgem, and I'll pick another. So I'll still have to linearize each one. Let's go fast here and just kind of pick one. Okay, so now I have 2 different clones, 3 inserts, so I should have 6 different clones that are created. And I'm going to rename this. And it will, again, the wizard will assess it. It'll say, yes, we're ready to clone, total of 6 clones made. And save out my primers. And that's it. And just let it run. And it'll go through and make those 6 clones, and it annotates them so we know where the different pieces are. And there it is. So for some users, very handy. If there's any questions at all with batch clone, I'd be happy to take them before I move on to a few genomics things.
No questions on cloning at this point. I do see a couple questions coming in on things for SeekMan and Megalign. I think we'll follow up with people after the webinar so that you can go ahead and show those genomics features, Matt.
Okay, great. All right, yes, I know we're getting close to the top of the hour, so this last bit won't take quite as long, because again, we're going to dive deeper into the genomics workflows in the next webinar for certain. And where most of the changes were done, there's a couple new workflows. I should say they're refurbished workflows from a few versions ago. And I'm just going to launch SeekMan Engine, and we can look at some of the new workflows and how they're labeled here. Okay, so SeekMan Engine, this is the front. If you launch it directly, now we've only launched SeekMan Engine from within SeekMan Ultra to this point, and so it starts out, a couple pages in, because you've already specified which workflow you want. But if you go to a new assembly, then you kind of see the full list of workflows. And so there's a couple of workflows that we brought back that are new for 17.1. One of them is this combine and reanalyze existing assemblies. The use case for this is, if you're doing, for example, RNA-seq, and you've got lots of different RNA-seq runs, and you chose a certain gene expression detection method, and then you decided, well, I want to change that. I want to use the same assembly. I don't want to reassemble all my data. I just want to reanalyze, or maybe add another assembly. In some cases, an assembly fails or something's wrong, so you want to add one new assembly. So it allows you to combine and reanalyze existing and not having to, rerun all those assemblies. And so the input then is assemblies. So I'm going to add some assemblies here. And in this case, these are some little RNA-seqs that were done with DE-seq2, I believe, which is on bioconductor for for differential gene expression. And you can see that when I add it, it recognizes this is RNA-seq. There was a replicate name applied. And so I'm just going to load 2 of those. And then the controls, which are labeled WT. So I'm just going to kind of show you how you might use this. We won't run the whole thing, but just so I can see the wizard says, okay, you've got you know, some replicates here. We have replicate names. They're all RNA-seq. We can go to the next page. It's already recognizes that these were used as, you know, with replicate sets, so they've been grouped together. So the RNA-C have been grouped together into a RNA-C replicate set, the wild type into a WT replicate set. And the control is recognized, it's already marked. So really it's because I'm redoing something that's already gone through the wizard, it recognizes that. And it's saying that this was done with DE-seq2. So for example, if I wanted to change that and use EdgeR, I can now choose EdgeR, next, you know, and this would be an EdgeR. And what it's going to do is take those assemblies and just recompute the gene expression values, right? Without having to reassemble them. With E. coli, it doesn't make that big of a difference. The assemblies themselves only take a few minutes, but some RNA-seqs, if you have a 20 sample RNA-seq, you know, it could take an hour or more for each sample. So then it's, you know, a full day of assembly you can save yourself by doing this. So it's a nice feature for users who are already generating data. Okay, so now I'm gonna just, so that's the combine workflow. So I'm gonna use the start over just to go and show you another workflow that is here, and that's under variant analysis resequencing. We brought the viral host, oops, I might have gone too fast there. Viral host integration detection. The tooltip says locate prophage and retroviral insertion sites in the host genome. So it can be used to find where prophages are in bacterial genomes or where retroviruses are in mammals. And in this particular workflow, you provide the 2 different templates and your data set, and it will automatically do a couple different assemblies together. So you could do this on your own if you knew exactly how to align to the one genome, export the reads, you know, align to the next genome, export the reads, that sort of thing. But this makes it a lot easier to find those sites, to find those reads that contain both viral sequences and host sequence, so those chimeric reads at those insertion sites. We have and again, I won't go through the whole workflow here, so I'll just mention that we have this, but we have a workflow page on our website that will explain this in more detail and provide more information if you have questions on it. So 2 smaller workflows. What I really want to talk about in the last couple minutes here is the de novo assembly and finishing. With version 17.1, we've introduced a couple of betas, and that's for long read de novo assembly. And so we have PacBio Nanopore de novo. This is for bacterial genomes primarily, maybe smaller eukaryotes. And it has a whole set of algorithms that we've developed for doing this de novo assembly. If you have short read data, you can also do a polishing. So I'll just kind of show you what that looks like. And it's really easy now to set this up. So we've, again, through the wizard, I can add my data. So here's some MinION data. A little bit of information. So we want to have an expected genome length. I can assemble all the reads, or more commonly, and what the default is, use a subset. So just use the longest, presumably best reads to achieve, you know, a certain depth, right? So I'll just stick with the defaults. We have an option for some data sets doing a, and I'll leave that tooltip up, to do a pre-correction, correct prior, can improve results, but it's not the same for all data sets. So there's some, you might try this with and without to see if it improves your assembly. And now I'm adding the polishing. So these are the short read sequences that can polish the output from the long read assembly. So I'm gonna load some Illumina data here. And I'm just trying to show you that we've made this very easy for users to set up, long read with Illumina. It's not separate actions. It's just a single action of with correction, assembly, and then polishing at the end. And here's some polishing options. How much data do I want to use for the polishing? How stringent do I want to make the alignment of those short reads? So you can kind of fine tune it and maybe have an initial pass at 1% and then use a genome finishing, take the same reads at a higher percentage. And again, that's something we can cover in the next webinar that focuses in this area. And we just pick the output, and I'll just get to the last page here. And again, it does an assessment of what you're trying to do. So this is long read de novo. It's going to use some RAM. It's also going to use some disk space. So it'll give an estimate of how much is needed and then what's available. And so we can run this on this computer, or again, run it up in the cloud if you're limited with your hardware resources. So very easy project setup for long read. There are some also, I'll just comment that there are some finishing, and this is new terminology in the wizard. And so we're offering, and as Katie mentioned at the start of the webinar, we'll have anywhere from a completely de novo assembly to you've got a draft genome and want to align data to correct errors in a draft genome, to refinement where you've got a high quality genome and you're really trying to scrub out the last few SNP-like differences. And so we're presenting multiple different options for folks to work with their data sets, maybe you try both. You might try de novo, aligning to a reference, and then additional tools and intermediate finishing steps that can guide the process. And again, that will be the August 5th webinar. We'll go into this in detail to show how to use these different workflows for genome finishing. And with that, I know we ran a couple minutes long, but I'd be happy to take any questions. And if I don't hear from you, I will try to answer all the questions that we get that we haven't gotten to today and get back to you via email. Thank you.
Yeah, thanks, Matt. I'm just going to share my screen. I know we're a couple minutes over, so if people need to leave, go ahead. I wanted to put up these URLs. If you want to see the release notes for Lasergene, that's up on our blog. We also have a lot of different workflow-based videos and tutorials. So if you go to DNASTAR.com/workflows, you can see some of the workflows Matt showed today, as well as other workflows in the software with some specific videos and tutorials. And then all of our user guides are on our training page, so DNASTAR.com/training. So I'll just keep these URLs up. And Matt, maybe we could take just a couple questions. If people need to go, that's fine.
Yeah, absolutely.
We have one question from Jody. Can you explain what you mean by the DSeq2 assembly file? I thought DSeq2 analyzed gene expression levels from raw read counts.
It does, yeah. And so what those were RNA-seq assemblies that have been run through the SeqMan engine wizard, and DESeq2 was chosen for the gene expression calculation. So what the wizard does is runs the assembly and then sends the information up to the cloud, which is running a instance of bioconductor with DESeq2, and that calculates the actual differential expression. So, and then it returns it back to the project. And so those projects had the DESeq2 calculations associated with them. And so what I demoed is that if I wanted to change that and say, well, I really want to use EdgeR, I can take those same, I don't have to reassemble everything. I can just load those assemblies in the combined workflow and just choose a different differential gene expression calculation.
Okay, great. I think there's a couple other things that are a bit outside the scope. So if you had a question and we didn't get to it, I'll follow up with you after the webinar. And just the last question that came in is if there's going to be a link to the recorded webinar, and yes, there will be. So we'll get that up on our website likely by tomorrow, if not by the end of the week. So look for an email from us with a link to the recording. And if you have time, there's a brief survey at the end of this webinar. We'd love to get your feedback. And just lastly, thank you for joining us. We hope to see you again at our next webinar. Take care, everyone.